Okay, today in biotech class we were chosen by Iowa State University to do an experiment for a local farmer. Local farmer had this problem where one of his 4 dogs had been chewing up his boots, and he had no idea which dog it was. So he gathered some hair that was left on one of the boots, which he assumes is hair from the culprit dog, and he packaged that up. And then he took samples of hair from all 4 dogs and sent those into Iowa State. Iowa State extracted the DNA and then passed everything on to Eagle Grove's biotechnology class, which you can see here is a wonderful looking group of students, and asked if they could figure out maybe what's going on here. So they started the experiment today by taking each sample of DNA and adding some restriction enzyme to it. Restriction enzyme basically breaks the DNA into smaller chunks and allows us to run it on electrophoresis gel. So right now they're adding a restriction enzyme called BGL1 to all the different DNA samples. Here you see one of the students using the micropipette to take the very small amount. I think it was like 3 microliters of restriction enzyme they have to add. 3 microliters is barely a drop on the tip of a pen, and so they're using very small quantities. And so in order to make sure things get mixed up, they have to run them through a microcentrifuge, which makes sure it spins it really fast and makes sure that all the liquid gets pulled down to the bottom of the microtube that they're using, and that's what they're doing right here. After they get the restriction enzyme put in to the DNA, they have to put it in a water bath at 37 degrees for 10 minutes, and that allows the DNA to be broken up into chunks. Now they're taking some migration dye and putting that in with the DNA samples. The migration dye will allow them to visually see the DNA moving along the electrophoresis gel, so they'll know when to shut it off. So again, here they're adding migration dye to their DNA samples. This is what a happy student looks like right here. Now, once the migration dye is added to the DNA samples, they have to load them into electrophoresis gel. These gels were prepared the night before. Basically, it's a gelatin made of agarose, and there's little holes at the end that they can load each sample of DNA into one of these different holes. And so that's what they're doing right now. They're getting the DNA samples into the micropipettes and then injecting them into the gels. You can see the wells here, and you can see that it's a dark blue color because of the migration dye. That's going to allow us to visually watch the DNA as it moves along the agarose gel. Once the gels are loaded, they have to add a 1x TBE buffer solution to the electrophoresis setup. This will allow the electricity to be conducted from the negative electrode to the positive electrode. DNA has a negative charge to it, so it's going to be attracted by the positive electrode. So they have to make sure they have the gel sitting in there with the proper orientation, or the DNA goes backwards and falls off the gel. This is what the setup looks like now. They have it hooked up to a power supply at 150 volts. They'll let that run for about an hour, and you'll see the DNA will move from the negative to the positive electrode, from this end down to this end, and that migration dye will move right along with it. When the migration dye gets to the very end here, that's when we'll stop it so it doesn't go off the edge. And then tomorrow we'll stain it, and we'll be able to actually see what the different samples of DNA look like. We'll have a spread of the culprit DNA, and then we'll have a spread of the other 4 dogs, and whichever one matches up to the culprit DNA, that's the culprit. That's the dog that's chewing up the boots. So that's our CSI lab for today, called DNA fingerprinting, compliments of Iowa State University.